Thank you very much, Maris, for this nice introduction. And I would like to welcome all of you who are interested in this topic, which is the topic of my research activity since 20 years or even more. So I am going to, first of all, I have no potential conflict of interest. This is the outline of my talk in a short overview on the etiology of male infertility. Then I'm going to talk about non-genetic factors, about genome-wide association studies as exome studies, and at the final part I would like to dedicate to male infertility versus chronic diseases, whether there is any genetic link. So next month, in July, we are going to celebrate the 40 years anniversary of the first IVF baby. And since that time, there are more than 7 million babies born from IVF techniques or later on from the ICSI techniques. And in about 50% of cases, we have a male factor. Male factor, which means the most frequently that there is a problem with sperm parameters. And whenever recently we reclassified the etiological factors of male infertility or male reproductive impairment, we actually classified into 4 major categories: hypothalamic pituitary axis disturbances, quantitative spermatogenic disturbances, qualitative spermatogenic disturbances, and ductal obstruction or dysfunction. And in each category, we emphasize the presence of genetic factors, which you can already test for them. And then we have the acquired non-genetic factors. And we introduced also a novel subgroup, which is called presumed genetic, because we are referring to idiopathic cases of quantitative or qualitative spermatogenic impairment. How much is important the genetic, the presumed genetic factors? If you look at this paper, which is a pilot study on the first young adult ICSI offsprings, as you can see, there is a significantly higher incidence in the ICSI offspring of oligozoospermia, asthenozoospermia in respect to natural pregnancy offsprings. So it's another hint that also the idiopathic cases are more likely part of them due to hidden yet unknown genetic factors. And it would be important to know what we are transmitting to the next generation by these techniques. So before then I talk about the genetic factors, the non-genetic factors, I would like to emphasize that our field, andrology and male reproduction, is quite complex. When you talk about azoospermia, you see it is a very homogeneous phenotype. It means that there are no sperm in the ejaculate. But apparently it is a homogeneous phenotype because we have the possibility of an azoospermic man with normal spermatogenesis at the testicular level, or we have, even for the non-obstructive cases, we have the possibility of a testis histology of hypospermatogenesis, very few normal tubules, and the vast majority are not normal. We can have Sertoli cell-only syndrome. We can have spermatogenic arrest, and the spermatogenic arrest can be arrested at spermatogonia, spermatocytic, spermatidic stages. And just to complicate the picture, we also can have complete and incomplete forms of these SCOS and spermatogenic arrest. So this is just to say you that whenever you think it is a homogeneous phenotype, it is not. It is a very heterogeneous phenotype, and we expect that for each of these histological forms, we may have different genetic factors, different genes are contributing to these histological pictures. So what is known in azoospermia? Azoospermic men, if you do the complete diagnostic workup, then you will end up with about 25% of them with known genetic factors. And these genetic factors are mainly karyotype abnormalities. In about 14% of cases, we have Klinefelter syndrome. Then we have Y chromosome microdeletions. It's about 8–10%, depending on the selection of the patients. A, B, and C. Mutations in the congenital hypo-hypo candidate genes. We can have androgen receptor mild resistances. CFTR mutations in congenital bilateral absence of vas deferens, and also TEX11 mutation. I'm going to talk about that. This is an X-linked gene. On the contrary, on the oligozoospermia side, we have less frequently the chance to find genetic factors. We have only 10%, and these are mainly karyotype abnormalities. And here I have to stress that mainly these are structural alterations, inversions, or translocations. This is important because the phenotype of these structural anomalies are actually oligozoospermia more frequently than azoospermia. This is why in the guidelines we are always saying that you should do it below 10 millions of spermatozoa, the karyotype analysis. Because in your patient it is balanced, but in the spermatozoa it can become unbalanced. So it is quite important to diagnose them. The Y chromosome microdeletions, the AZFC deletions, are typically associated with with severe oligozoospermia. Partial, very rarely, we can find partial AZF-A and B deletions and the GR-GR deletions. And then again, some mutations, mild mutation in the androgen receptor and also the CFTR gene mutations can be associated with unilateral agenesis of vas deferens. And in that case, you can have normozoospermia or oligozoospermia. So I don't want to talk about karyotype anymore. I just would like to make an update on the Y chromosome diagnostic test. As you all know, in 1976, Tiepolo and Zuffardi introduced the concept that on the long arm of the Y chromosome there would be an AZF region. Then Peter Vogt, at the molecular level, defined the AZF-A and B region, C deletions. And then in 2002, when the Y chromosome sequence became available, David Page laboratory defined these five recurrently deleted regions, and they discovered that the B and C region actually are overlapping. We all know that this kind of deletion, these complete deletions, or A, B, and C, they have a clear-cut cause-effect relationship with impaired sperm production, either with azoospermia or severe oligozoospermia. In 2000, we proposed that besides the diagnostic value of this test, we also can use this test for prediction for the testis biopsy. And we proposed that the complete A and B deletions have a testis phenotype which is basically incompatible with the sperm production, so there is virtually zero probability to get sperm in the testis biopsy. SCOS or spermatogenic arrest in these two cases, and the AZF-C region deletions are more heterogeneous phenotype, both in semen, it can be associated with azoospermia or severe oligozoospermia, or in the testis phenotype level, hypospermatogenesis or even SCOS. And in this case, the average frequency of finding spermatozoa is about 50%. So this is the reason, the concept about complete A and complete B deletions. This is why in the EMQN best practice guidelines, we actually stressed about the importance of defining the extension of A and B deletions for prognostic purposes. And this is just an example for the B deletions. As you can see, you have the routine markers, then the extension. And here, at that time, in 2013, we suggested that you have alternatively either SY143 or 1192. But then in 2016, Stoops and ourselves also with Frank Tuteman, we found that actually these are not alternative markers. You have to test for 1192 because there are cases which are positive for these markers, and in this case, you can find spermatozoa in the testes. So now we are working on this topic, and we are even more defining molecularly this issue, because it is important that although it seems a complete AZF-B deletion, but depending on the markers you are using, you may now change the prediction, because it is very important to do this marker. So the clinical significance of complete AZF deletions are actually clear-cut cause-effect relationship. Testing indicated in less than 5 million spermatozoa per ml, but I would like to call attention that the large majority, almost all of them, has less than 1 million spermatozoa. There are no association with cryptorchidis, hypospadia, testicular germ cell tumors. I'm talking about the complete deletions. Then the prognostic value, I already told you about the 192 markers, work in progress, and I hope in a few months' time we are going to publish an amendum to the guidelines about these markers. And then it is well known that if you have a 45X cell lines in even at low mosaicism, it has a negative predictive value for sperm retrieval. So you should also perform the karyotype and look for low frequency of these cell lines. Obligatory transmission to the deletion of to the male offspring, and we cannot really predict the exact phenotype, the semen phenotype of this offspring, but we can say that there will be no normozoospermia because never these deletions, the complete deletions, were found in normozoospermic men. Some years ago, some authors raised the questions about risk for general health for those men who are carrying the Y deletions. And this study in 2011 was actually claiming that AZF deletion carriers with normal karyotype had 10% of them had Shox deletions or Shox haploinsufficiencies. So we did a big multi-center study, and actually our conclusion was that since we didn't find any of these 225 deletion carriers with normal karyotype, we didn't find any Shox haploinsufficiency, so the conclusion was that there is no risk for these kind of diseases. Very recently, in human reproduction, this group also raised the question about behavioral, emotional, and psychiatric disorders in patients with terminal BC deletions. And it is interesting. I mean, in the AZFB region, you can have genes which are expressed in the brain, so theoretically, it could be also possible, but I think that we need more data before we start to worry about it. So I think that these are examples how it would be nice to have multi-center studies and with specific questions like this one. Now, the last 15 years, we were actually dealing also with another big question. The question was whether the partial deletions, the partial AZF-C deletions, and especially the GR-GR deletion, what are their role in male infertility? And these deletions remove half of the gene content of the AZF-C region. And this is the largest European data we have. This is from my lab. We published it in Human Genetics, then we updated. This is from Journal Medical Genetics, from Fundació Puigvert, from Elizabeth Ars laboratory. And so we put together this data, and as you can see, with 1,000 Spanish and Italian patients and controls, we ended up with an odds ratio of 4. It means that a carrier of the GR-GR deletion have a 4-fold time higher probability to be severe oligozoospermic or moderate oligozoospermic, because these kind of deletions are more frequently found in moderate and severe oligozoospermic men. And this was a very large multi-ethnic study from all over the world, from Rosen, and he also demonstrated that there is a double risk for spermatogenic failure. Now, in the clinical setting, whenever you have a risk factor, I know it's difficult to deal with it because you don't know exactly what to say to the patient. We test for it in Florence, but there are also many other laboratories who are testing for it. And I would say we can consider it as a cofactor for reducing spermatogenic efficiency. It's like when we find a varicocele, when we find a recurrent infection. So it's a kind of genetic factors which certainly worsen the situation, but not enough to be the sole cause of an oligozoospermia, because it can be found also in normospermic men. Now, there were studies claiming that perhaps the partial AZFC deletions predisposes to complete AZFC deletions. And why? Because the substrate for the deletions are homologous sequences on the long arm, and these B2B4 called amplicons, whenever you have a partial deletion, they are closer. And during meiosis, the distance is lower, so in theory, you can have a higher chance to have a deletion due to the non-allelic homologous recombination. And there are papers about haplogroups with constitutive partial deletions among complete AZFC deletion carriers, and we used to test the father of our patients with complete AZFC deletion, and we found one father who had GRGR deletion and the son had a complete AZFC deletion. So it means that, again, it is something to do with multi-center study to find out whether we can consider GRGR deletion as a pre-mutation, because it's actually closer the amplicons, and whether we are talking about transmission of a potential transmission of a proven genetic cause like the complete AZFC deletion. So for starting from GRGR deletion carriers, whether it is something that can lead to complete AZFC deletions in the spermatozoa. Now, after the Y chromosome, I would like to go to the X chromosome, and the X chromosome, for me, it's a perfect target for male infertility for two reasons. One is because David Page Laboratory has published that at least 10% of its gene content is specialized in male germ cell development. And there is a higher than expected number of genes which are expressed in the testes in genes or linked to the X chromosome. So we started to talk about masculinization of the X chromosome. And then another reason why I like this chromosome, because mutations are in hemizygous in the male. So if you have one mutation, you have no normal allele to compensate, and if the mutation is pathogenic, then you would have expecting to have direct manifestation of the phenotype. And what has been done, in 2012, we did a RACGH study on the whole X chromosome, high resolution X chromosome, and then we did this follow-up study. And in this study, we found the CMV67, which is on the which was found only in patients with spermatogenic disturbances. We also found a potential risk factor which was not confirmed in a Czech population, but the CMV67 actually was confirmed in a recent study from China. And in this study, they are also claiming that this deletion affecting the proximal copy of a cancer testis antigen. There is full of cancer testis antigens on the X chromosome, and we know that they are expressed exclusively in the testis physiologically, but we do not know what they are doing there. So there's a lot of question marks, but these are testis-specific genes. Another big shot was in 2015 in New England Journal of Medicine. We had this paper about TEX11 mutations, and this is coming from the US and from Münster. And the other study in the same year from David Page and Jeremy Wang Laboratory is both of them were actually stating that these kind of mutations are more frequently found in meiotic arrested patients. And I must say that in this study also very nicely they demonstrated that in the mouse this gene is absolutely necessary for the meiosis, which would fit with the human phenotype. But I must say that you have to be careful when you look at this gene, because also missense variants exactly in the same exons, when they reported in the infertile men, but also in fertile men, you can have missense variants, and they are indicating that you have to be careful when you attribute to a mutation a pathogenic role. So certainly it is a very interesting gene, and I would say the deletions, the frame shift are certainly important pathogenic mutations, but for the missense you have to be careful. Now, after that I have talked about the quantitative spermatogenic disturbances, I would like to talk about congenital hypogonadotropic hypogonadism. And just to recall you some notions about the physiology of the hypothalamic pituitary axis, we know that the DNA, the GnRH neuron genesis occurs outside of the brain. These neurons have to migrate together with the olfactory neurons to the brain, and then when they arrive to the hypothalamus, they have a number of upstream signals, and then they produce the GnRH, and then it goes to the pituitary FSH, LH productive cells who have the GnRH receptor. Now, there are at least 35 candidate genes which have been reported in relationship with this disease, which account for approximately 40% of cases. And although we can clinically, we can distinguish between the syndromic and the non-syndromic forms, because syndromic, it means anosmia, hyposmia, or other non-reproductive malformations and so forth, so on. But at the genetic level, we have a number of genes which seems to be overlapping between the two manifestations, the two clinical manifestations. And we talk about the fact that from a genetic point of view, not always you can distinguish between the two kind of manifestations. There is another peculiarity of this disease, the oligogenicity and degenicity. It means that all these 35 genes, they may have one heterozygous mutation and 2 or 3 genes are carrying heterozygous mutations. So sometimes it's not follow Mendelian inheritance. And another very interesting thing is that the 10% of these genetic disease are actually reversible. It means that after testosterone replacement therapy, which we use to these men to give, they can recover the hypothalamic pituitary gonadal axis. So periodically, we used to interrupt the testosterone treatment and see whether the axis has recovered. And in about 10% of cases, you will see it. I would like to advise you, to suggest, to read this very nice paper, the consensus statement from 2015 for further information on this pathology. I also would like to mention about another hypogonadotropic hypogonadism, but in this case, it's affecting only the reproductive part, and especially the FSH production. The chair of this session, Marislan, is an international expert on this FSH beta polymorphism. If you have a TT genotype, you will have about a 50% reduction of your capacity to produce FSH. And she and her collaborators nicely demonstrated that whenever you have this TT genotype, actually it is also associated with lower testis volume and lower sperm concentration. It looks like that there is an inappropriately normal low FSH level, and we could say it might hypo-hypo, somebody is calling functional hypo-hypo, affecting spermatogenic efficiency. And it has a potential for pharmacogenetic treatment. So we could actually think about giving FSH to those men who are genetically unable to produce enough FSH. Now I would go to the next etiological category, which is the qualitative spermatogenic failure. And I would like just to touch two teratozoospermia types. So these monomorphic teratozoospermia, these are recessive diseases. So the large majority of these studies were actually reporting consanguineous cases or familial cases. And we know that they are very rare, but they are important to be detected. We have these two genes, the aurora kinase C for the macrozoospermia and the DPY19L2, which is the major candidate gene for globozoospermia. And why they are important? Because they are associated with repeated fertilization failure. Even when you do the ICSI, for instance, with globozoospermic men, You will have problem with the oocyte activation. There are epigenetic problems with this sperm. There are sperm DNA fragmentation higher level. And on the other side, the macrozoospermia is associated with tetraploidia. So it is important, they are very rare, but if you have it, you can actually diagnose, in the majority of cases, the genetic background and be careful by using these spermatozoa for ICSI. I am not going to talk about ductal obstruction and dysfunction because I already mentioned the CFTR gene mutations. I would like to spend some time on the idiopathic oligoazoospermia, which are actually the large majority of the quantitative spermatogenic failures. It's about 40–50% of cases. And as I said, we presumed that they can be genetic of origin. And during the last, let's say, almost 20 years, this was the way how to explore the human genome. And as you can see in our little field of androgenetics, we also started to make array-based genome-wide association studies, and starting from 2013, you can see the first studies on exome and the next generation. So what we found in the SNP arrays, so these studies were done on idiopathic infertility, and with the general thought that male infertility is very common. It's about 7% of male have a problem of spermatogenesis in the general population. So common disease, common polymorphism. But in our case, it did not work out. We have 4 studies, 2 from the US, 2 from China, and there were no overlapping genetic risk factors in these 4 studies. And there were only in the Chinese population validation studies for these 15 confirmed SNPs, and that's it. So we don't know what to do with these SNPs. From a clinical point of view, of course, they are not usable, but it is possible that in the future, maybe the combination of low size effect risk factors may have a role in, let's say, in oligozoospermia. I would not predict that they have any role in azoospermia, but in oligozoospermia eventually may have some role. In this study, you can see that they evaluated the combination of 4 SNPs. If you have only 1 or 0, this is the sperm number. If you have 4 SNPs, the bad SNPs combined in your patient, then you will have a lower sperm count and a lower birth rate. These are hypotheses. This is something to go on and to see with the 15 SNPs which were confirmed in the previous studies. So the genome-wide genotyping risk factor search was quite unsuccessful, and now we would like to show you some data on the NGS or high throughput studies dealing with exome and gene panel, which would have, in theory, a higher clinical impact. We are looking for rare variants in these cases. If you go to the PubMed, you will find about 49 articles from 2013, and they are monomorphic teratozoospermia. They are all, let's say, almost all are familial cases of monomorphic terato, astenozoospermia, familial non-obstructive azoospermia, congenital absence of vas deferens, and some syndromic diseases. And they are all recessive. Either you have homozygosity or very rarely compound heterozygosity. It means that in heterozygosis, these genes which have been successfully demonstrated, but in heterozygosity they are compatible with sperm production. So these genes are useful to know which they are, but they causes male infertility only in homozygosity. So we also started with the exome analysis some years ago, and this is just an example of a consanguineous, so from 2 brothers with consanguineous parents. They have spermatocytic arrest, both brothers. We did the recessive model. We prioritized after the we were looking for shared homozygous variants, prioritized for functional effect, for potential role in azoospermia, And just very briefly to show you that we found this RNF212 mutation in exon 2, and this protein is important for meiosis. And luckily enough, we also have the knockout data already published for this gene. And as you can see, it is important for chiasm formation, and in the mouse, there is a loss of spermatocytes. And actually, in collaboration with Willy Barends in Rotterdam, we also did this meiotic study, and we demonstrated that our brothers recapitulate exactly the same phenotype as the mouse. So this is those rare cases when exactly the same phenomenon we have in the mouse and in the human. So in this case, we were able to make the diagnosis of the spermatocytic arrest. So I would say that if you look at the literature, exome studies are quite successful in identifying the genetic cause in familial cases of infertility, but the problem is that we have the large majority of our patients, at least they say that they are coming from non-consanguineous families. So work is in progress. Missing etiology is under study. We have this large international consortium, the Gemini, and we also have a consortium of genomics of male infertility with Joris Weltmans, Tuttelmann, and also others who are setting up a large consortium to share information between different laboratories, because we really need to join our data. We will have the presentation of my postdoc in this afternoon, the whole X chromosome gene panel study, and if you are interested in it, you can join at the cold room later on. So, last slides I would like to dedicate to an emerging issue and emerging topic, which is spermatogenic disturbances in general health. Why I am talking about it? Because there are 5 epidemiological studies which are claiming that male infertility is associated with higher morbidity and lower life expectancy. And as you can see here in this USA study, if you have more than two sperm parameters, altered sperm parameters, then you have a lower survival probability. Then there were bioinformatics studies also. A study is made a single method disease analysis, and in these studies you can see that again infertility seems to be associated with a number of cancers, including also lymphoma and malignancies in the urogenital tract. Then Don Conrad, also with his colleagues, made this genetic intersection study between the MGI male infertility genes and the cosmic cancer genes. And they also found a 5-fold enrichment of cosmic cancer genes in the MGI male factor infertility list, compared with genes that are not on this MGA list. So there are epidemiological and bioinformatic clues that there could be a correlation between genes involved in spermatogenesis and something related to cancer and general health. The most obvious genetic link is, of course, the Klinefelter syndrome, because in this syndrome we have hypoandrogenism, azoospermia, sexual dysfunction, but due to hypoandrogenism. But we also have the extra X chromosome, which is likely to be responsible for the pathological conditions which are associated with Klinefelter syndrome. And we have also osteoporosis and metabolic syndrome, which is probably, which are related to both the hypoandrogenism and other X-linked factors. And here again, we have some specific cancers also associated. So Klinefelter syndrome patients must be followed up for all their life. We don't have to tell them just take the androgen replacement therapy. They have to be followed up during their lifetime for all these diseases. Now, apart from Klinefelter syndrome, when we did the RA comparative genomic hybridization studies, 2 studies are coming from whole genome. We did the X chromosome and Yatsenko, so the X chromosome specific. We had one common finding. And here now I can show that there is a poster from Marisland group, first author is Punab. They also found what? They found that what we also published before, that there is a higher deletion load in infertile versus normal zoospermic men, which is especially evident on the X chromosome, at least from our study and from 2-10-month study. So we hypothesized in PLOS ONE in 2012, when we published this data, that the excessive loss of genetic material in patients versus control could be a tip of an iceberg, which actually linking somehow some of the patients with high CMV load between infertility and general health. So that could be a potential other genetic link to cancer and to chronic diseases. Now, last discovery we did, it was a whole exome sequencing in a patient affected by non-obstructive azoospermia, here the sertoli cell-only syndrome. We excluded known genetic factors, and the parents were first cousins from Andalusia. This is a patient, and this study we carried out in Fundació Puigverd with my postdoc, and this was a very interesting case because when we filtered the mutations, the variants, and we ended up with 380 homozygous variants, and from these we prioritized those which were predicted to be damaging with our in-house pathogenic index, which is based on prediction tools, of course. What we found this FANCA missense mutation, which is in homozygosis in this patient, and its pathogenicity was proven by functional studies already because this mutation is known. So after this case, actually, we did a more extensive study looking for our SCOS patients. So, as I told you, NOAA, it's a heterogeneous situation. You have to know the testis histology in order to be able to be more precise in your diagnosis. So we took the SCOS patients with hematological parameters which were similar to our patient with SCOS, and in whom we found this homozygous mutation. He was not having anemia, because Fanconi anemia is, of course, is normally associated with anemia. He had borderline values, and when we selected these 27 patients with similar blood parameters and with the same histology, we found an additional man with compound heterozygosity for the FANCA mutation. And that would mean that if you are very selective and if you know the hematological parameters, actually you have to ask for blood count in case of sertoli cell-only syndrome, because you can find this occult Fanconi anemia. This is about 10% of Fanconi anemia patients who have occult Fanconi anemia, which is coming out late in their age, and very often the diagnosis is made because of a cancer. So in this case, we have the first cases where we were able to diagnose Fanconi anemia before cancer, and it was because of the sertoli cell-only syndrome genetic analysis. And just 3 days ago, it has been published in Genetics in Medicine, and if you would like to further discuss with me, I will be in front of the poster this afternoon, and I am happy to answer to your questions there. So in summary, concerning the infertility and chronic disease issue, I think that we have known genetic causes, which we know that they are associated with chronic diseases like congenital hypo, Klinefelter. We have the pleiotropy for these congenital hypo issues. But now we are talking about the possibility that defect in DNA repair may affect both the bone marrow and the spermatogenesis, and we are talking about genomic instability due to high deletion load. So this is my last slide now to summarize that genetics in andrology, I think it's very important. As I told you, 7 million babies were born by ICSI and IVF. We should know what we are doing when we inject the spermatozoa. We should know what is behind the impaired sperm production, so the diagnosis is important. The transmission, to know what we are eventually transmitting, if we're transmitting a genetic disease, the prognosis for TESA, especially the Y chromosome deletion studies. And then we have this issue about prevention, the reproductive health versus general health. I did not talk about interaction between genetic background and environment because there are not very much data about that. And also, of course, an emerging issue is epigenetic, which is, again, in our field is emerging. Pharmacogenetics is another interesting topic, and there are some controversial results in the literature. Also ourselves, we did some study, but it is something that I think will be developed, and the FSH beta and FSH receptor polymorphism are very interesting in this sense. So if you would like to talk about genetics of male infertility and listen to it, we are going to have the European Congress of Andrology, and we have a lot of genetic talks also during this Congress. So if somebody is interested, please come to Budapest. And thank you for your attention.